Good morning, everybody. It's my pleasure to welcome you all to this 15-day certificate course internship on computer-aided drug and vaccine design, which we are planning to organize between 29 January to 12 February 2022. Before going into the session, I would like to give you a brief introduction about the Institute of Innovations. Institute of Innovations, we are a registered enterprise in MSME, Government of India. We are located in Tiruvannamalai. We are doing activities related to science and technology. Our activities include scientific collaboration, project outsourcing, technical assistance, and more activities we are doing. So we had organized various events in the past. From May 2020, we had organized several events. You can able to see the details of the events here. We had organized International Online Summit on New Disease Outbreaks, International Online Colloquium on Prospects in Bioenergy Research, 10 days internship program on nanotechnology, 6 days weekend online training course on clinical research, clinical data management, and clinical data science, 15 days internship on petroleum industry waste management, 15 days internship on IoT and renewable systems. We had organized a conference called International Conference on Advances in Science and Technology during 2nd and 3rd April 2021. So these are the glimpses of our events, various events. So we are more particular in selecting our resource person for all our events. All of the resource persons are experts in their own field. We are selecting the experts from not only from India. We are approaching the persons who are working abroad and asking them to give their expertise lecture in this in our events. So this is the next event. So for the internship program, we had around 18 invited lectures. So this is the course we had organized in 2020. After overwhelming response from audience and requests from most of the colleges, we had organized this course again. So this is the conference brochures. So this is the second time we organized the clinical research course, and we are planning for the clinical research course in coming months. That is in the from the 3rd week of February, we are starting another batch. We will be uploading the information to you later. So we had organized this certificate course on computer drug and vaccine design during June 2021, 16 to 30 June 21. That course was organized daily from 6 to 7 p.m. But due to the time restrictions, this time we are organizing 3 hours per day in weekend. So after that course, we had organized a short course on drug design for SARS-CoV-2 and precision medicine design. It is a short course for 5 days. And then we organized basics of oncology and cancer stem cells. This is a short course. Then we organized an internship program for petroleum industry waste management. This is the second time we organized. And last month we had organized a certificate course on fermentation technology. After that, we had organized an international webinar on computer drug design. Most of you will know about this webinar. And after this, we are now into the 15-day certificate course on and internship on computer drug and vaccine design. So this course we are planning to organize between 29 January to 12 February. In fact, the classes will be on 5 days. That is on Saturday and Sundays, 29, 30 January, 5, 6, and 12 February. So the course time will be 10 a.m. to 11 a.m., 3 p.m. to 4 p.m. and 7 p.m. to 8 p.m. on these Saturdays and Sundays. So for this particular course, we are having Dr. P.K. Praveen Kumar, Associate Professor from Sri Venkateswara College of Engineering, Sri Perumbudur. He is a resource person for our course, and he is a TAR Fellow, DSC is a TAR Fellow in IIT Madras. He is best friend of me, and he readily accepted and agreed to handle this course. I feel that Dr. Praveen Kumar is a correct person to handle this course because in our previous version of this course, we had received a very good feedback for this course, and most of the participants are praised us to organize this course, and I am sure that this course will be a milestone in your career, in your studies, because you are going to study more about drug designing and vaccine design. That is, which is the limelight of this particular time. That is, after COVID, this drug and vaccine design came into limelight. So, in this short note, I would like to welcome Dr. P.K. Praveen Kumar to take over the session without any further delay. Thank you, sir. Welcome, sir.
Thank you, Professor, for providing me one more opportunity to present on the same 15-day certificate course and internship on computer-aided drug and vaccine design. So, with no further delay, I'd like to share my PPT of today's lecture. Can I, Professor?
Yes,
Thank
sir.
you.
Sure, sir.
Good morning to you, and as well as the participants here. May I know how many participants enrolled here, sir, totally?
Here there are actually 64 participants are here. Okay. 73 participants enrolled in the course, and some participants have asked me that they are having class today morning. They'll be joining in the afternoon session.
Okay, Professor. Okay, thank you. So then I think I can switch off my video now presently
Yes,
for internet streaming, better internet streaming. So good morning, participants. Actually, I need a kind of a full interaction from your side. So unless without interaction, just like a lecture, it won't be good. And I can tell my strategy. I'll be taking nearly 30 minutes or 25 minutes lecture, and remaining 25 minutes hands-on session. The last 10 minutes for questions and any kind of a thing you like to discuss. Okay, so this is how I plan. Okay, 50 minutes session, 10 minutes for questions. Okay, so even you can accommodate any questions if you have anything. Okay, and you can ask me later, because there is a lot to explain. Lot actually. For the past I think last June month, I have taken this kind of a course, online certificate course on computer-aided drug and vaccine design for Institute of Innovations. And in HVC, myself, I have conducted the same kind of a course. This is a 15 hours course. Actually, the major topics what we are going to take is my screen visible? Can anyone interact with me? Is my screen visible?
Sir. Yes, sir.
Yes, sure.
Yes, okay. Thank you. Actually, we broadly classified into 3 topics. One is drug design, and the molecular mechanics, and the third one is the vaccine design. Okay, so these are the 3 major topics. Initially, I like to take the drug design. So today we'll be taking about this. Okay, so about drug target identification using pathway databases, drug target modeling and validation, and drug likeness properties, drug databases, drug ADMET properties. Okay, and tomorrow we'll be seeing the remaining 2 more drug design. Drug design and molecular mechanics are associated with each other. Okay, so whatever you do practical exposure with online software tools and databases, the same, you try to learn the theoretical concepts in molecular mechanics. Of course, in drug design, we come across the QACR techniques, docking techniques, dynamic simulation, everything comes under that. But actually, there is a lot to know about theory. Even if you study about a disease, the biochemistry of a specific disease is important. I mean, biochemistry of a protein, of a drug. So this also is very important, along with an immunology. Okay. So if you are studying microbiology, biochemistry and microbiology is important. Of course, cell biology involves in all the streams. Okay. So if you come across not only this, the bioinformatics also comes in GATE, Bioinformatics National Certification Exam. There is one exam called Bioinformatics National Certification Exam. So it is equivalent to GAT. Okay. So for all these examinations, there is also a need to know about all this. If you see GAT, they are not asking anything more particular to the bioinformatics syllabus of an analyst. They are also asking mostly related to drug design. So even I think for your master's program, I mean a master's entrance examinations, or even presently you find numerous opportunities in informatics. Okay, even I found nearly 4 to 5 companies are asking or they're providing salaries more than 10 lakhs per annum in the drug design areas. Okay, so whatever I'm going to take, okay, so this is just a 15 hours course. I'm also not just restricting up to drug design, also taking up to the vaccine design. So I'm taking few more concepts, but there is a lot to know about this. Okay, so I'm just taking a part of version. Okay, you have a lot of thing to know. So I take 25 minutes theory session, 25 minutes hands-on session. Okay, if you miss any sessions, try to watch the recorded video sent by Professor Praveen sir. There is no way. Okay, if you miss one topic, you know, each and every topic is interlinked with each other. If you miss one session, the understanding of this session is difficult. You understand? So this is what I want to take initially, because you should know the strategy of a course instructor, how he is going to take. Okay, and you can follow according to that. Okay, even if you can't able to grab it on a single time watching, you can watch for multiple times. Okay, until you try to get an output from your system. So I suggest everyone immediately after the lecture, you can practice and see. Then only you can get an real-time exposure. You can get a hands-on experience with it. Because if possible, I see that because the 15 hours is very less. If possible, I can also ask the participant to share their video and do something in online. Okay, if you interact like that, even it will be better for me. Okay, instead of simply showing some kind of a result from my screen, if some volunteers or interested to display or you to learn or explore the skills, you are welcome. You can share your laptop, but you should have a high configuration. Not a high configuration, I mean it's a 4 GB RAM. Okay, so for practicing the tools, most of the tools, whatever I'm going to explain and all, whatever biological databases, okay, I'm not going to take any kind of commercial softwares or commercial kind of a databases to explore for studying this kind of a certificate course. Mostly it is a free online softwares. Okay, so everything is possible with you now. Okay, not everything will limit up to the theory. This is a pure hands-on experience session. So of course it's a kind of a workshop, or we can also call it as a certificate or an internship course. Okay, I provide all this, of course. So of course after acquiring these concepts, okay, I see that I can mingle these two concepts in a single session on a last day. And the last day, and the last session, you have a quiz. Okay, so whatever concepts I'm going to teach, okay, you will be having the form of just for knowledge testing. Okay, so I think there won't be any problem with certificates, it seems. Last time also it was assured like that. So just for your knowledge. Okay, so test your knowledge. Okay, this is important even for job interviews. Okay, so now I think I can go ahead with the first chapter, that is about drug design, target identification using pathway databases. Yeah, so now we can go ahead. What is drug discovery? So I have heard some terms like drug discovery, drug design, drug development. What are these areas actually? What do you mean by, what is the difference between these three terms? So you remember what I told just now? One is drug discovery, the other is drug design, the third one is drug development. Can anyone can unmute and answer? Do you have any idea? So if you don't have an idea, I just brief it out. Drug discovery, you find, okay, drug design as well as drug development. So these are the two areas covers under drug discovery. So you have various computational areas in drug discovery. So the can be in modeling and simulation. Yeah, I also have a few more areas. Okay, in drug discovery. Now we see how about the drug discovery. Actually, if you see in drug discovery, okay, as I already told, you find basically whatever we call it as a discovery is what the design. Okay, so just the design alone will give you a drug. You should develop the drug. You should develop the drug by testing in vitro. So whatever you do in drug design is in silico. Actually, there are three studies in we do in lab. One is in silico. The in silico part of work is what drug design. Then what is drug development? For checking the drug, for developing a drug, you need in vitro studies as well as in vivo studies. Okay. And before going to the explaining these concepts, I like to know, I like to interact with your participant because myself, I think Professor Praveen sir has introduced myself. Okay. So can I get some kind of an interaction from you? Like how many, I like to check how many are B.Tech and how many are M.Tech and how many are research scholars. Can I start interacting in the chat box now? So, so list of UG students. Okay, so kindly type your names in the chat box. List of UG students. So I can check out the numbers. Okay, so anyone from other than biotech, anyone other from biotech now, you can stop typing. I understand. So there are plenty in. So any other from biotech, you can type your name as well as the other department because there are multiple disciplinaries, biochemistry. Thank you, Mallikarjun. And who else? Any other streams? Okay. Now, only PG students can type your names. Only PG students. I think in UG, plenty of students enrolled, including in SVC. Only PG students? Okay. Good. So, even I can find biochemistry, MSc biochemistry. Any MSc bioinformatics? MSc bioinformatics? Chemistry, biotechnology streams or that. Because I want to know how much you understand. Some are already proficient in informatics, B.Tech bioinformatics, M.Tech bioinformatics, PhD. I don't know. So I like to check. Okay, zoology, microbiology is there. Good, good. Okay, how many are PhD scholars? Now only PhD scholars can type your names. So Kamini will have. Good. So okay, so only one. Okay, okay. So okay, thank you for your introduction. Good. So now I think I can go ahead with my explanation. So I was explaining you later to drug discovery and development. So I was explaining you like discovery basically uses in silico approaches. In silico, we mean we study in computational. Okay, in silicon. In silico means in silicon. We have silicon chips in CPU, isn't it? The microprocessor. So whatever we study in silicon is that's what considers computational. Is a drug design is a part of drug discovery. And development, you go for in vitro. In in vitro, we mean we do experiments with tissue culture flask, okay, test tubes, etc. Okay, and if you go for in vivo, that is a third study which we do with preclinical studies with small animals generally. Okay, so only in veterinary colleges they do with large animals. So we generally do with small animals like rodents, rats, mice, hamsters, rabbits. So all these things we also do. We have an excellent animal house facility in SVC. So we do experiments with all this. Okay, so not only this, we also have something called drug delivery. Okay, the drug delivery can only be tested in preclinical as well as in the clinical studies. Okay, so the after the development, the delivery process is there. Okay, that's not mentioned, of course. In the subsequent slides, we can find this. Okay, now if you see the target, what is the need? Why we have to go for a kind of a drug discovery? So the need is like if a drug has to come to a market, okay, it has to take minimum of 20 years. Okay, that in 20 years, for the computational part alone, see here they have taken nearly 3 years. Okay, nowadays this target discovery, that is the computational part, is reduced very less than a year. Okay, so some can complete within few months. Okay, like a UG project. So how it can be done? So the even whatever you discover drug, okay, don't limit up to the talking and simulation. There is a lot more techniques to validate your results. If you develop a kind of a protein, I mean, you design a kind of a protein 3D structure, you should validate a protein. If you have a compound, you should validate that kind of a compound. Okay, all these things has to be done. Then only you have to go for talking, simulation, using network pharmacology. You have to study the interaction of different proteins with the same drug. So all these things are very important. Okay, if you want to study a specific disease, if you want to target a specific disease, or not only testing has to be done, the validation has to be done. Okay, so this course, of course, provide you some kind of a basics related to testing as well as validation in computational studies. Okay, so it depends. Okay, if you work day and night, you can complete within a year. Okay, even computational studies nowadays. And because in drugs, okay, you find billions of compounds deposited in various biological databases. So if you want to test, if you want to kind of a design a drug, okay, it's not, don't limit up to a plant or some few compounds. You can also take that kind of a compounds, okay, and go for visual screening. Then you can take a best drug, okay, and you can do a fragment based screening. Then you can identify a best kind of a drug, a lead development from the drug. Okay, so all these things are there. Just like the taking a drug and going for finding a receptor, talking, and simply telling, I found out a drug for a disease. No, it's not a kind of a good a researcher kind of an idea. Okay, so we should develop a kind of a strategy. You should have a plan for a drug discovery. Okay, so only I am telling it also take 1 to 3 years. Okay, later if you see screening, okay, that is a screening lead development. See here they have taken up to 5 years. So whatever I told, okay, to not simply restricting up to a plant or is a 2 or 3 plants like what you do in Ayurveda, Siddha, Unani, homeopathy. What we do, we take a group of plants related to a particular disease. For example, for corona, so we take what kind of chorna or what kind of need. So yearly in 2019, so in Ayurveda, what they prefer, can I get interaction from the students? Only through interaction we can make this course better. Okay. You want to make best, try to interact. Well, so what's the general kind of an Ayurvedic drug or herbal formulation generally the doctors prescribe at that time? Because we don't have a proper drug in 2019. So if anyone affected with COVID-19, okay, they take ivermectin, they take a malarial drug, they take even HIV drug. I don't know how HIV can able to, HIV is integrated with a kind of a coronavirus kind of a proteins. Okay, so near 8 kind of a drugs. Okay, so some kind of an antiviral drugs the doctors prescribe and they put a kind of a medical bill up to 4.5 lakhs, 6 lakhs based on the type of hospital where you admit the patients. It's very huge, and even you can't able to even the patient survival rate is very less at that time, isn't it? So generally, in home remedies, we came to know in the in the end of 2019. What is that? What is a herbal formulation? What your neighbors have taken if they got infected with COVID-19? What is that? 11. What is that? Very good. You are right, man. So Kabasura Kudinir. Okay. So earlier, okay, if for most of the viral diseases, for a dengue all this we go for Nilaveg. But for this, the best one is the Kabasuira Kudini. So taken from more plants, isn't it? So they take a kind of an herbal formulation. That's what prescribed. And we of course go for steam inhalation to avoid such a kind of a problems. Okay. So one is COVID-19 has provided a lot of impact in the biotechnology. So many projects got funded, and also some you can find the biotechnology is blooming with other fields, like it's emerged, it linked with IT, CSC, EC. So many people are also linked, yeah, in drug discovery and development for even the infectious disease COVID-19, in the form of disinfectants, in the form of a kind of mask preparation, in the form of so many devices came, isn't it? So all these things happen only the IoT devices, all these things came with uh, um, mingled with the health sector. Okay, so if you see the last 15 years, okay, we generally go for preclinical trials for a single year. Okay, we can check with animal studies, we can develop humanized mice models. Okay, so we can go for a nude mice. So we can do all this kind of a testing, and later we can come across with what is the best drug. Okay, we screen from the computational studies, we test with these animals, and we conclude a few kind of drugs. And whatever drugs we have identified from preclinical trials, we pass it for phase 1 to phase 4 clinical trials with humans. Okay, so the phase 1 to phase 4 clinical trials are tested, the drugs are tested with humans. Okay, so I think the students from fourth year can able to understand, because all these things you studied in biopharmaceutical technology. I don't think the third year, second year have this kind of a basics. Okay, and there is one course called biopharmaceutical technology where you study these concepts in detail. Okay, and also there is one more a large subject, large field called toxicology and pharmacokinetics. Okay, so the B form, M form study pharmacokinetics, pharmacodynamics, etc. Toxicology is of course a very important kind of a study. Okay, whatever drug you develop, we have to check the toxicity. Okay, so we have also toxicity prediction kind of a databases. Okay, so whatever we have to check, even for the preclinical, some kind of a databases are developed, and some more databases are developing, like whatever you want to check with animals to minimize usage of animals. Okay, to avoid sacrificing the animals, even some databases are existing to check the efficacy of the drug, to check the dosage of a drug. Okay, even if you want to fix a dosage, efficacy, bioavailability, so whatever you call, okay, various pharmacokinetic terms. Okay, so whatever factors you want to check with animals, we can check with the computational tools itself. Okay, so only I'm telling within a year, okay, we can able to even complete the animal studies. We can test the drugs with the animals, and later we can take a few more years to check with the human. So only after 20 years, okay. So earlier it was up to 35 years. Now 20 years, and I think for the past 5 years I can able to find even the drugs are discovered within 15 years. Okay, the proper approval. So whatever I am telling, the approval of drugs from FDA. Okay, FDA. What is FDA? Can someone expand this term, FDA? So only the phase 4 clinical trials, okay, we call this as post-marketing surveillance, okay, PMS. Okay, so in that stage only, the FDA it gets involved for approving the drugs. Okay, if FDA
Free trade agreement.
what is it, madam? Exactly.
Free
Federal?
trade agreement.
No. Someone?
Drug administration.
Exactly. You are right. You are right. Food and Drug Administration. Okay. So this is the body. Okay, approved body who approve the drugs. Okay. So the US people can take, India people can take, any other regions can able to take. Okay, if FDA is approved. Okay, and there is one more approval body from India. What is that?
IP.
What is that? IP. IP. You're very good. So that is an Indian pharmacopoeia. You are right, Param. And US people generally follow US pharmacopoeia. What is that pharmacopoeia? What do you mean by pharmacopoeia? And US follow US pharmacopoeia. India follow India pharmacopoeia. What do you mean by that? Can anyone know that? Can you explain about that? For a drug, we have some guidelines. Okay, so how to use, what to use, and how to prepare, manufacture drug in the industries. That's like that. The industries can't involve in a manufacture of a drug. It's not like an Ayurvedic formulation or Siddha formulation. So if any drug, so the drug, whatever I'm telling, is an allopathy drug. Allopathy, ALLOPATHY. The what we take from the medical stores. Okay, in dispensaries we get drugs, isn't it? So whatever drugs we take, we purchase our the allopathic form. Okay, so the allopathic drugs has to satisfy. Okay, there's the FDA kind of an approval. The one in FDA approval, you can find the drugs is marketed. But if the drugs has to come to the market, obviously it has to follow this kind of a guidelines. So good manufacturing practices. You heard about this GMT? Okay, if you want to test the drugs, okay, with animals, okay, we have to follow good laboratory practices. Good manufacturing practices we follow for drug manufacture. Good laboratory practices, we follow the guidelines for testing the drugs with the animals. So these are some of the guidelines we should know. Okay, and in India, other than these guidelines, okay, there is an approved body like an FDA. FDA is a universal approved body. Okay, but in India we use this CDSCO. Can someone expand this term? Or within this short time, I might have got from internet. Didn't get the answer. What is this? I have provided you the answer. Just you have to give me the expansion of this term. What is this? If not,
Central Drug Standard Control Organization.
Very good. You are right, madam. Good. So that is the approved kind of a body. Okay, from India. If serious approved kind of a drugs, we can directly take. Okay, so with no hesitation. Still, some drugs like paracetamol. No, not paracetamol. What is it? So yearly we had aspirin, isn't it? So aspirin and Vicks Action 500, Sinarest. These are the popular tablets. Even in childhood, I will be taking these kind of tablets. These tablets are banned because of side effects. So whatever, even the drugs are approved, okay, just because of adverse side effects yielding later related to cardiovascular disease or kind of hepatological kind of a disorders. Okay, so these are the major kind of a disease, including neurological. Okay, the neurological kind of a disease. So just because of these kind of a side effects, okay, some drugs even after approval they got banned. So obviously that will be removed from the list. Okay, these kind of drugs are not suggested, but some even in the drugs in the market you find these kind of drugs are less marketed. Okay, so these kind of drugs. So to know about the adverse drug reactions, okay, and about the drugs profile, even we have computational kind of a databases. Okay, we have computational databases even to find the adverse drug reactions. Okay, so all these things are very important, isn't it? So before checking with the animals or with the humans, you should study everything. You should start from a scratch, okay? And you have to screen billions of drugs and you identify a best drug for a target receptor of a disease. For a disease, you have multiple protein receptors responsible, but you should also identify which is the best protein receptor, okay, best protein target. So based on the disease target, only you have to screen a drug. isn't it? So this itself, it's a very huge task, isn't it? If you try to check with animals, billions of animals has to be sacrificed. So to avoid this, okay, so the computer originated in the 1950s, 1960s, 1978, 79, the internet came. Okay, in 1980s, the www get popularized, and the databases got emerged from bioinformatics area from 1980 onwards, and in 1990 you find even system biology just started, and in 2000 you find the genome kind of databases are developed. Later in 2010, okay, synthetic biology came into existence, and a few more kind of databases, including network kind of databases developed. Okay, there's a within a span of 50 years, you have large areas of development in computational field, especially in drug design, it can be a vaccine design. So all these things, we whatever we developed in silico, we have reduced cost, we have reduced the time for discovering a drug for a disease. So these are some of the areas of modeling and simulation. Metabolism modeling, manufacturing modeling, protein structure, homology discovery, kinetic modeling, function prediction. So all these things will be seen in subsequent lectures. So now we can come across our first kind of the types of drug design. So if you see the types of drug design, I simply broadly classify it as traditional drug design and structure-based drug design. So if you see the traditional drug design, okay, so generally the drug, how a drug is designed in 1970s to 1990s. So that's what we generally call it as a traditional drug design. So in that age, okay, in that era, what is happened? So they generally take a plants or a combination of a plants for a particular disease. They grind it using the pestle and mortar. They take in the form of a churna, or they take in the form of a paste, or in the form of a syrup, herbal syrup formulation, something like that. Okay, so they take a group of compounds, isn't it? So what exactly they take? They take a group of compounds from a series of plants for treating a particular disease. So they're not bound to a single compound specific to a plant. Okay, that's what a traditional drug design. So that's what generally in Ayurveda, Siddha, so whatever they deploy the strategies the same. Okay, we call it as a traditional drug design. So whatever compounds, like if you take even Kavasira, Kudinate, okay, so they simply go for a because we don't have a proper approved drug for COVID-19 nowadays. If you see for Omicron, okay, a lot of memes came that Dolo 650 is sufficient. Okay, even if someone is approaching the dispensaries, give me some kind of half kg of Dolo 650. Give me some 5 kg of Dolo 650, something like that. Okay, some kind of a memes are coming, isn't it? For Omicron, simply the paracetamol is sufficient to treat that kind of a disease. Along with the paracetamol, of course, you have to take drugs for cough, sore throat, cold, etc. Okay, so I've taken just 2 tablets. Okay, nowadays even some memes came like, even if you're not infected with corona for the up to present, you must be an alien. I don't know. Even that's what happening on. So everyone got at least 1 or 2 days infection, if not prolonged up to 10 days or 15 days. Okay, everyone got infected nowadays. That you should accept. Okay, so just a drug, okay, paracetamol, along with so what I have taken, actually I have taken a drug, so that actually cured me for a cough, that is cetirizine hydrochloride for sore throat, cough, etc. Okay, so even if you get infected, I think I suggest you take this kind of a tablet. Okay, these 2 tablets are sufficient. But now if you take a delta variant, alpha, beta, gamma, so many variants are there for a coronavirus, isn't it? So up to 10 variants are there, and for each and every variant, I'll be discussing all this in subsequent lectures. Okay, I'm just giving an outline. Okay, like we have nearly more than 10 variants, and even today I got 11th variant from China, where 1 out of 3 patients definitely will be going to die. Okay, now a deadly strain emerged from China. I mean, a deadly strain of coronavirus. I don't know when it is going to impact India. So, up to the lifetime, okay, even Dr. Soumya Swaminathan, WHO chief of our India, also provided the information that even up to our lifetime, the coronavirus is going to come along with us. Only thing is that you have to learn how to live with the virus. Okay, so nowadays, not only for the job opportunities, even it is understand for better health living. Okay, for better safety. Okay, even you should have to know what drug exactly you can use for a kind of a disease. Okay, so you can design your own drug in your system. Okay, so the traditional drug design, just like going for carbapenem, codeine, nilavibranor, codeine, so whatever you take, okay, so that's the kind of a series of compounds after biological testing. The biological testing, I mean in vitro studies. Okay, so in vitro studies, you have to develop a few compounds from the combinatorial chemistry labs, medicinal chemistry labs, and if the compounds yield promising results with in vitro, then you have to pass it. If the trials are promising, then you can pass it to the preclinical studies. Okay, so then we can go for the second based drug design, that is structure based drug design, where you're not involving a group of compounds from multiple plants, but you take a selective compound, a single compound from a plant. Okay, and if possible, you can link with one compound of one plant with the second compound of same plant or second compound of another plant, and you can take if both the drugs give a synergistic effect, then you can take the drug for treating the disease. Okay, so you're not taking a group of compounds, but you're taking a selective compounds. Okay, so how you take a selective compound? These compounds are selected based on the kind of binding studies with the target receptor. Whatever ligand you take, okay, the ligand is generally, if you see in drug design, we use two important terms. One is ligand, the another is receptor or target receptor, however you call. Okay, the ligand is generally a drug, inhibitor, or we call that as a drug or inhibitor. In pharmacologist term, we call it as an agonist. Okay, if it is a drug, we call it as an agonist, 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 and if it is an inhibitor, we call it as an antagonist. Okay, so these are the terms, various terms we use it for a ligand. Okay, ligand term in drug design. The another is a target or a receptor. Generally, it's a protein, diseased protein. We should not target a normal protein for finding the drug efficacy. You should check with the disease protein. So, of course, the 3D structure of the disease protein has to be determined. Then, whatever drug you have to screen, you should do the binding studies. Okay, after modeling, then that's what we call it as a docking. Then you have to go for checking the binding study stability with respect to time using simulation studies. Okay, modeling, docking, simulation. So this is the strategy we have to use. Okay, step by step, modeling, docking, simulation. These are the major studies we involve in drug design. So after finding the kind of a drugs, okay, we synthesize the drugs in medicinal chemistry lab and pass it for in vitro studies. And if you find promising results, pass it to the preclinical studies. That is checking the drug efficacy with the animals. So drug discovery and development. So discovery, as I mentioned, it's an in silico studies, and development we go for in vitro as well as in vivo. In vivo, I'm not simply restricting up to the preclinical. I'm also taking the clinical studies. Okay, this is also important, isn't it? This is what I told, phase 1 to phase 4 trials. So generally, we check with phase 1 to phase 3 and phase 4. Of course, it has moved to the market. Okay, so when it has moved to the market, when it has moved to the dispensary, if it has properly approved by FDA. What is EMA? you find one more term, EMA. Someone can expand this term, EMA. Because I found PG and PhD scholars are there. Even you can interact with it if you know. What is EMA?
So it's European Medicine Agency.
Very good. You are right, madam. Okay, so like this, even you can, if you know, you can answer, or you can find from the Google. Okay, then you can make the subject interesting. Very good interaction from you participants up to present. But now, if you see the strategic research, okay, so we generally go for target discovery. The target discovery can be done through target identification, not just identifying the disease protein is sufficient. You should validate the protein. Even if you model the protein, you should validate the protein. Of course, this is an important step. And then you can go for developing the drug. The drug can be developed through exploratory research. Okay, so that is through hit generation and lead identification. So for this, various screening technologies used, virtual screening, assay development. So assay development, this can be done in vitro. In vitro. Okay, we have so many in vitro development assays. Okay, so you can use immunofluorescent assays. So you can go for FACS kind of an analysis, even with it, with the cell lines, whatever you have. Do with kind of a cell lines, okay, or cells, whatever you have. High throughput screening, again, a competition. Drug design is a competition. Hit to lead, again, competition. Okay, so this covers the exploratory research and candidate drug selection. So lead optimization, a part comes under, this comes under actually computational, as well as you can check with in vivo. Okay, so computation, you try initially check with the computation studies, then you can check the same with the animal models. In animals, you can check the same ADME, efficacy, safety, etc. So we can't simply rely on the computational technique. Sometimes it may fail. If it fails, then you have to check with the animals, isn't it? So that's a one-day validation. And even if the drugs prove successful results with the animals, may sometimes it fails in clinical trials. May it fail in phase 3, even if it clears phase 1 or phase 2. That is based on various factors, okay, based on various properties of the drug. Okay, so all these things are very important in drug discovery and development. So as I already mentioned, in drug discovery, okay, also the biological assays, okay, you should know about even the best drug discovery scientist has to know about ELISA. You has to know the hyper-through screening using microarrays. Okay, so wet lab techniques. I am just linking the computational techniques with the wet lab. So you have to use ELISA techniques. You have to use microarray techniques. Okay, so whatever computation you have checked, you should try cross-validate with these techniques. And there is also for the drug receptor binding studies, you can use these studies. Circular dichroism spectroscopy. This is important to find how ligand effectively binding with the receptor. So these are the important kind of techniques. Of course, cloning, the PCR analysis, etc. Of course, also it plays an important role. If you go for RNA kind of therapeutic targets, okay, I mean, so you know, the drugs can also not limited up to a plant. You can also take drugs from animals, okay, or it can also be like RNA, biological molecules, macromolecules like RNA, DNA, okay. So all this also also works as a ligands for a specific targets. Okay. Now, if you go for drug development, okay, so whatever you discovered, okay, through the computational techniques, kind of like a structure-based drug design, we just go for using various kind of chemistry, biology, even physics involved, okay, to check the best drug, okay, identified for a disease target, okay. Then whatever you have done, okay, you have to develop using the preclinical and clinical trials. And drug elucidation, this is the third part. In drug elucidation, okay, so you should check for example, if a person is infected with Omicron, okay, Omicron kind of a coronavirus, and how it can be just a single drug, paracetamol, Dolo 650 can able to cure? No, the Dolo 650 is not used for simply curing. It's just for reducing the fever, to brought down the temperature alone. It helps. Then, of course, body pain you can bring down. But how about cough, sore throat, cold? You should use some kind of an antibiotic, isn't it? Okay, so you can use as I already mentioned, cetirizine or amoxicillin. So these are the antibiotics generally the doctors prescribe. And I found even some doctors are prescribing ivermectin. I don't know. Only from my student I came to know yesterday that she is taking the ivermectin. No, ivermectin is generally used to remove the worms. I I don't know even why the doctors are prescribing this. Okay, I prefer the Dolo 650 and taking an antibiotic. Okay, amoxicillin or even cetirizine. Okay, hydrochloride. So if you take these two tablets, Dolo 650, ivermectin, how about the synergistic effect? Whether one drug is going to give some kind of an adverse effect, or even if I take Dolo 650 with the cetirizine after my lecture, whether it gives any kind of side effect. That's important, isn't it? It also depends based on the patients, based on each and every patient have an unique HLA genes. So not all the drugs set, okay, for all the patients. That's what we call it as precision medicine design. You know, from 2010, this is the term, okay, which is very popular over the world. Okay, so nowadays they're not the patients are not taking, like even for a few dispensaries, even in my college. Okay, you know, if someone who goes for any kind of like I am suffering from fever, or I am suffering from cold, I am suffering from cough, I am suffering from body pain, or I am suffering from even loose motion, so they are giving some kind of SAM tablet. How a single tablet is curing all the diseases, we don't know. Okay, that's a very rude kind of and crude kind of an approach, isn't it? So this has to be overcome by the precision medicine. Okay, so some drugs is effective for some patients. Okay, so this kind of a drug elucidation by studying the synergistic profiles of the drugs, a combination of drugs, what you take for a particular disease is important. Of course, genetics, okay, as I told, HLA genes vary from person to person. If it is a normal fever, cold, cough, no problem. But if it is a kind of a disorder, syndrome, okay, or a kind of a rare disease, so what exactly we can do? One is by studying the genome. One is studying the genes. Okay, even it can be a cancer or autoimmune disease. Studying the genes information is important. Okay, to target the specific disease using precision medicine approach. So the drug elucidation is, of course, an important one. Okay, and participants are actually, I'm going to provide you this kind of lectures, okay, through in the form of a materials. I provide you the materials. Okay, so I have framed for all the 15 chapters. Okay, so I'll be posting, or I'll be passing it to Professor Praveen sir for posting in the Google Classroom, however he like. Okay, so you can learn this. So these are some of the theoretical concepts. Okay, just 5 minutes remaining. So if you have any doubts, you can ask me now. So any doubts you can ask me. In the next lecture, I go ahead with the continuation of what I left. Like I basically restrict my theoretical part because now I explained you more theory. So I go with the target identification using pathway, okay, and validation, modeling, etc. in the next session. At what time? 3 to 4, isn't it? So at that time I can explain you. So do you have any doubts? You can ask me, or even the 5 minutes I can take my session for practical. Any doubts? You can unmute and question me if you have. Or can I go ahead? Please explain docking. Docking will be in the 9th or 10th lecture. Today's first lecture, isn't it? So the docking is simply a binding studies. Okay, high throughput screening will be saying in 5th lecture. Okay, fourth or fifth lecture. So we'll be seeing in each and every lecture all these computational techniques you can find. But before that, you should know also some basics. Okay, so like you should know what is the source of generally ligands. So now I will be telling you the ligands source. NCBI PubChem. So the biological public domain databases, you know, NCBI, we call that as National Center Biological Information. We have plenty of databases with more kind of a data here. PubChem is one of the database where it's a repository of chemical compounds. For example, if I want to find for aspirin, it's also there in the bottom. See, some drugs are there in the bottom. If you want to find a paracetamol, okay, so you can also search like this. If you find aspirin, you might have clicked there, it has reached you just like this. So paracetamol is a brand name, but generic name is acetaminophen. Okay, so everywhere if you call acetaminophen in the world, they can immediately got it. Okay, so acetaminophen, you can find one base compound, but you also find some similarity compounds of acetaminophen. So here in NCBI PubChem, you don't simply restrict up to a single compound. Whatever compound you search, you find similar compounds related to a specific compound. And if you want to do kind of an high throughput screening or visual screening, whatever you call, okay. So visual screening is of course a screening of multiple drugs to identify a best drug. To identify a best drug from a combination of a drug, so you can download this. This downloads all the 84 results in a single data set. Okay, so 84 compounds are downloaded in a single file. Now, if I want to find this specific drug information, so not only this NCBI PubChem, you find the drugs, you also find a repository of natural compounds, some synthetic compounds. Okay, you can find these compounds in the form of 2D structure, in the form of also 3D structure. Not only find 2D and 3D structure information of chemical compounds in the NCBI PubChem, but also you can find the physical and chemical properties of these compounds and the biological properties. So what exactly this compound is used, okay, and how about what are the types of diseases it can cure, okay, all this information. If you want to find the 2D structure of the compound, okay, find here, simply click download, okay, to download simply the acetaminophen and paracetamol structure. If you want to download the 3D structure, click download, you find STF. STF, this is a 3D structure generally I prefer to download. You have various models like ball and stick model, wireframe model, space filling. These are the types of models we can find for the chemical compounds, drugs, and also for the proteins, we have an exclusive database. Okay, whatever I'm showing is for the ligands. Okay, so also you can find the canonical SMILES format. This is one more format. Okay, INCHA format. So these are the various formats. And if you scroll down, you can find physical and chemical properties of the drug. Okay, so more details about this we see in the third lecture. Okay, so this is a repository. Okay, NCBI PubChem is one of the small molecule database. Okay, like NCBI PubChem, okay, you can find CHMBL. Okay, it's also a manually curated database of bioactive molecules. Okay, this is also a public domain database. You know, NCBI, the origin is from US. And CHEMBL, of course, you can come to know this is a European database for chemical compounds. Okay, nearly 2.1 million compounds are there, and how about the assets? So if you want to search a specific compound, like how I search paracetamol there, okay, search for the compound here like this. Okay, so I can better go for aspirin now. Aspirin, okay, GM type 1, aspirin. Okay, search for the drug, you find this kind of a drug here, aspirin, and some kind of related compounds down. Like CHMBL, we can go for drug bank. Okay, so I'll be completing within 1 or 2 minutes. Okay, so drug bank. In drug bank, you can find drugs like this. Okay, so you can search for aspirin, search like this. So not only in drug bank you can search drugs, you can search targets, that is proteins. You can search through pathways. So these are the different search fields it can take. Okay, instead of searching drugs, you can search for targets, pathways, indications, etc. So see, so many details. So this is a fully annotated database of a drug. So the name itself implies drug bank online. It doesn't limit up to already approved drugs, allopathy drugs. We also find some kind of natural compounds here. Okay, bioactive molecules we can find here, even in drug bank. We can study the pharmacology. See, this much information is provided. Okay, you can find products, category, chemical identifiers, targets, enzymes. So aspirin is targeting nearly 19 Hello, am I audible to you?
Yes, sir.
So I think I have completed up to drug bank, the small molecule database. Okay, so in the next session, we study about the target database, modeling, validation, etc. Okay, so do you have any doubts? You can ask me. Do you have any doubts presently?
Good morning, sir.
Good morning.
Sir, which assay is used to bind the ligand with the receptors?
We have plenty of assays. So for ligand receptor, protein receptor binding assay, as I already told, we can go for the drug protein binding assays. So we can go for ELISA, but generally the best one is a circular dichroism spectroscopy. So this is the only in this technique we can find more about the drug protein receptor interactions. So ELISA generally for cell-based assays, microarrays, protein-based or gene-based kind of an expressions you can go for. So my suggestion is to go for CD assays.
Yes, sir. Thank you.
Thank you. Any other doubts? Okay, participants. So thank you for the interaction. So we meet at 3 o'clock or for join 5 minutes earlier. Okay. So, okay. Thank you. Professor, are you available?
Yes, Professor. I'm here. Okay. Okay, participants, if you have any queries, you can make note of it and you can ask in further sessions also. The permission of Professor, shall we wind up this session?
Yes, Professor. So, as you have suggested, if participants post any questions, I can answer in the Google Classroom.
Yes.
How about the material, Professor? So, can I provide it to you? You might.
You can post it on the Google Classroom, Professor. No problem.
Yes. Because you might have if you want to post Institute of Innovations logo, emblem, and any other kind of modifications.
Yeah, that will be nice. I think if you provide, I can do that.
Yes, Professor. So I can give you the chapter of contents as well as the tutorial. Okay. Thank you. Thank you, Professor.
Yes, Professor. Thank you. Thank you, participants. You can sign off right now. I'll post you the feedback link up for this session. Just fill that feedback form so that you can improve further sessions. And please join the evening session at 3 p.m. I request all of the participants to join well in time before 10 minutes of the session so that it will be easy for us to finish the session on time. Thank you.